Class is now starting. All attendees are in listen-only mode.
Oh, can you guys see my screen?
Yes.
Okay, all right. Well, thank you all. Well, hi everyone. Thank you for joining us to another episode of Chat with Green Aggies. Today is April the 23rd. Again, if y'all have any questions, go to the Facebook page, Ask Aggie Experts. We have this chat with Aggie Grins. Just send us questions and stuff there. We'll definitely talk about those in the next chat. So today, before I talk about the new crops, I want to mention several of the webinars that we're working together. You know that I have been working on crapemyrtle bark scale for a couple of years, since 2014. By the way, Erfang is part of the team, so we're after so many years of research and everything, we've made a lot of progress on this topic. So we're offering not one, not two, not three, but 4 webinars, one per month. In May, it's going to be feed and preference on not only just on crepe myrtles, but also on beautyberry and pomegranate and other things. And then we'll talk about consumer grower survey in June. We're going to talk about mechanical control, biopesticides. In July, we're going to talk about the beneficials, the imported and the native. And in August, Erfang and possibly with Dr. Mike Merchant, we'll talk about chemical control. So this is a very holistic, systematic, strategies, all the informations that we have on the progress that we have made on control and managing a crepe myrtle bark scale. So all the information is on the Ask Green Expert there. So hopefully you guys can join us in those webinars, in addition to the weekly chat that we have here. All right. I forgot to ask you guys this in the poll. How many of you know this crop? And this is what I want to focus on today. For me, Jujube is a crop that is a fruit crop, and also a great potential as a landscape, edible landscape plant. And actually, I've already got some nurserymen in Texas who are interested in this. So what you see here, these are these are different Jujube cultivars. For instance, can anyone guess? Maybe just right now, not the audience, just from the co-presenters. Do you guys know what this what does it look like?
Mushroom.
Yay! Thank you. Well, there's still some Chinese in you. So this specific cultivar is actually mushroom Jujube. It looks just like mushroom. Okay, let's do another one, another test. What about this one? Becky, what do you think? What does it look like? There's a little baby song that I've been singing every night to my daughter.
Oh, is it the shark? Is it a baby shark?
No, not baby shark. No. And the music, the song sounds like da 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 da. Can you guess?
A teapot?
Yes, this is a teapot. This is a teapot. Well, this is
like a little shark.
The handle is on the other side. So yeah, this is 1 year Dr. Yao. She probably has some connections with Yao Min like 500 years ago. But anyway, Dr. Yao at New Mexico State University sent me these Jujube fruits, and that's how I got started with Dr. Yao. So these are some additional Jujubes. Just I put the quarter there just so that you could see the size. So a lot of the folks may not be familiar with this, but it's a fruit that's very crunchy, sweet, kind of like a dry apple, that kind of thing. The nice thing about this crop is that you can see that they are flowering now. So compare, and do you guys know when, like the rosaceae, like peaches and, apples, pears, when they flower? Early spring, right? About early spring. So jujubes flower during this time of the year. That tells you one thing. Think about the frost damage. Are we going to have any frost damage? Are we going to have any frost damage during this time of the year, when they flower? So, like for peaches, there's always a chance that, they flower and, all those rosaceae plants that they flower before they leaf out. For this, they don't have, the damage to flowers and thus to the yield, to the fruiting is very minimal. The branching of this thing is totally weird. You look at this thing, this is where the fruiting, the flowering branches coming out. It's totally weird. It's like, it has a cluster of things, and then just one of them, weird enough, one of them may just take off, and then, that's your New Year's growth. So, and, in addition to minimal damage to the flowers, to the fruiting, there's also advantage of this crop is that they have just few disease and bugs. Very drought tolerant once established. Actually, that's one of the reasons that Dr. Yao is promoting this fruit in New Mexico. Because of their water requirement. Okay, this is these are the plants that I have in the hort track that I'm doing a trial. We have about Dr. Yao sent me 10 cultivars that I'm growing, so you could see that, that's my hand. You could see the growth that they have, and of course there is always cultivar differences. Like this one, it's at least twice the size of my thumb versus this one is just about the size of my thumb. After 1 year growth. So when I send this two pictures to Dr. Yao yesterday, and she said, yeah, there's always this huge cultivar difference. And one of the cultivar that she worked on was so slow that she's already got papers published. And you know how slow that you could get, field trial papers published. She's already got field trial paper published on the other cultivars versus this one still in the establishment stage, and she doesn't have enough science to share. So like I told you that I have 10 cultivars, about 50 plants, hopefully that we're going to have fruits. And this is what I'm looking at. To invite you all to a yummy jujube tasting workshop sometimes in the future. So this is what we're looking for. Again, this picture is from Dr. Yao. She has many cultivars. Oh, another good thing is you notice that some of the fruits here are green. Some of these are like maroon red. Maybe I should name this instead of calling it jujube. Maybe just call it a maroon apple or something. But anyway, notice that some of these, so they could be picked. They have a long, because of low water content, they could be picked. The picking window is very long, from very green to very maroonish, very mature, and they start to taste sweet even at this young stage. So this is the crop that one of the crops that I'm working on. Hopefully that some of you some of you are would get interested in this crop and come to our field day. All right, next, I'm gonna, these are Dr. Yankee Joe's slides. Yankee, do you want to speak up?
Can you hear me?
Yes.
So I can add some jujube, and when I was young, back in South Korea, my parents have a big jujube tree in my yard, and it was very delicious. And actually, they dry out, and during the so they're mature in the fall, and then they're harvested and then dry out. They make a very dark, maroon color. and it can make a tea. It can make some kind of spice material. You can make some kind of ingredients. It makes it sweet, so you can add it to some kind of cooking with some other meat or chicken, and so it's really very popular, the fruits, and I have a good memory on the jujube. The bad side.
One of the audience commented that no, I assume you're a lady. She planted these jujube at a homeless shelter a few years ago. The Asian homeless love these fruits, but she hated the thorns, and she said that tastes like apple to her. So what, Becky, you nodded. Do you have any experience with jujube?
No experience really. I just know how much I have friends in the Asian community that love them. So I wasn't surprised to hear that they love them. So.
But the bad thing is the tree was here in my backyard was dead some year. It is caused by phytoplasm, like Candidata disease. It's insect vector, very serious. If you got infected, like slowly progress multiple year. The tree is not completely dead, but it's make a lot of witch's broom type of symptom develop, and there's no fruit, and it looks weird, and so there is no value of this tree. Once you got phytoplasms infected, so it's a great crop, but it's very. I mean, my experience is that the tree is not worth it with a phytoplasm infection. So, so be aware. I hope there is no phytoplasm in Texas.
The phytoplasm, I mean, that affects everything, right? Not just jujubes.
No, that is like a subspecies. There's Candidatus. There's I think this is related to the Huanglong bean in the citrus, and no, what is is a zebra chip?
Okay, I see what you're saying.
All right.
Okay.
Kevin Ong is shaking his head in agreement, I'm assuming.
So Kevin maybe add it later, but the phytoplasm is you can culture these pathogens, and it's transmitted by the vector, some kind of insect. I don't know which one is, and so
leafhopper.
So yeah. So hopefully we don't have those phytoplasma here.
You want to move on to your research?
Sure. So it's instead of like details my research, I just some common sense that are relevant to our work. So we probably know that good soils is promote health of plants, and so if you prepare good soils, good nutrition, and that provide healthy plants. If bad soils and the plants not growing well, also somehow they sick, some disease you find it. So I mean, those are kind of we've kind of promoted that make the good soil to make a plant healthy, and we know about that. We added good compost. We hopefully promoted some good soil. So we know already a lot of good ideas. I mean, a good feeling about good soil, but really, what is in a good soil? I mean, we know there's a NPK, or there's micronutrients in there that support the plant, but that's not all. I mean, you can add those essential nutrition in it, the plant still gets sick, or they didn't grow well. So we know there's a lot of microbes in the soil, right? And so, but which one is good, which one is bad? We don't know. And I mean, one spoon of the soil, and there is there's a lot of microbes in there. Even like, I mean, some of the information that said is almost like 100 million bacteria in 1 tablespoon of your soils in your garden, right? So there's a lot of organisms growing in there, and it can be good one, it could be bad one, you know. I mean, but it's anyhow, there is a lot of organisms in the soil, and so I mean, we can isolate them. We can we can take some soil sample, and then we kind of dilute it in the water, and then we can grow them in the artificial media, different kind of artificial media, see if we can isolate some interesting bacteria or fungus from it, and then kind of individually investigate how they work. That has been working many years in the microbiology world. And the good thing is, yet indeed we find some lot of interesting bacteria, fungus from the soil, and they have very effective, effectively control, you know, the plant pathogenic disease, and also it can promote the plants more resist to various environmental stress. So this is one of the examples that the one side was inoculated with the soil microbes, soil bacteria, and then the other side is just normal soil. So with this, if we inundate it, we grow a lot of this bacteria in the lab, we pour into the in the plants, and so the roots are kind of protected by this bacteria. And when the drought stress comes, so if you didn't water it, of course the plants got you know, get stressed and start wilting out. And but with this bacteria, the plants resist a little longer, increase the tolerance to the drought stress. So that's something that you know, good things, and we can potentially use this kind of bacteria. We inoculate a lot in the soil or around the plants, so the plants get little benefit. They grow better, or resist to some of the soil disease, and also it can help the plants more resist to the drought stress in this case. Okay, next slide. And so the other okay, so this is another kind of concept. So the first slide is we isolate it and we culture artificially large scale, and then you can pour into the plant so that the plants got better resist to many biotic and biotic stress. But as I mentioned before, there is so many microbes in the soil, okay, and even very few, very you know, small amount of the microbe actually culturable, isolate from, and then culturable in the artificial media. Majority of the microbes are cannot culturable, and they just work together. Okay, they maybe they work together, not a single isolate dominance. They just work together, and as a whole that makes some you know, good health, promote good health for the plant. So this is some kind of conceptual experiment to how this is numerous microbes in the soil. Without isolating, isolating anything, we can try to manipulate the whole microbes in the soil to make the plants more healthy or resist to the droughts in these cases. So this is something like a circle, like a generational cyclic selection of beneficial microbes from the soil. So you start with the number one. Can you see that number one there? So there's a plant. We isolate it. We collect from different soil from different locations. You plant it on it, and then number 2, you see they growing there, and then we exposed to the stress. So in this case, we didn't water it. So I guess a lot of plants got wilting and dead, and there is only like top 2% or 3% of plants are showing some, looks healthy. Okay, I don't know why, but it's something we assume that there's some microbes associated with it. So okay, we pick top 2% of the soil plants. Okay, we get together number 4, and then we got amalgamated. Okay, and then we added some additional media, soil media, and then we make the repotted as number 5, and then you plant again the same plants. So we're going back to the number 2, and then planted the same plants, and then again number 3 we exposed to the drought stress. Okay, and then again there's some plants dying, some plants looks healthy, so we pick only top 2% out of it. Okay, and then do the same thing, multiple cycle. So if it's multiple cycle, and depend on what kind of system is, if it's a plant growing fast and short the cycle, something like annual crop like wheat or corn, we can do these cycles pretty quickly, like a month each cycle. So after 5-6 generation of this selection of the microbes by host, actually you can create it or engineer the soil microbes particularly adapted to the drought stress, and so make those plants more resistant to the drought stress. Okay, next slide. So once you do the multiple generations of selection of the microbes in the soil, and we label as HMME, host-mediated microbiomes, engineer soil, you plant it, okay, and then compare it to the sterile soil, and definitely somehow this soil support the plant more resistant to drought stress. Okay, so we don't know what kind of bacteria or what kind of fungi, what kind of microorganism in there, but we kind of selections of the microbes what we want, and then promote the plants better. So that kind of indication that microbes is really important in the plant growth. Okay, so anything that we can promote the good health microbes that really very beneficial for different kind of crop systems. So current genomic study, genomic tools are really affordable and well developed, and so the sequencing is you just send any kind of sample to this genomic center service, and they can send it with three weeks. Within 2-3 weeks, you give your lots of gene sequence data that you work with. So very affordable to any kind of sequence of those microbes in the soil. So that give us scientists, microbiologists start to looking at without culturing this complicated microbes, we can actually understand what kind of microbes actually in the beneficial soil or the good soil versus what kind of microbes composition in the bad soil or the potentially promoting pathogens in the soil. So yes, still have a lot of study need to be done, but it's a really hot topic to understand some whole you call the microbiome. So all the based on the genome sequence, we be able to look at the snapshot of what kind of microbes are present in the given soil conditions or around the root of the plants we're interested in. So I think that's I mean it's how can we apply this kind of knowledge to the actually in the field is still that kind of a long way to go, but it's the way it is. We have more idea of this microbiome, and which is we can it helps we can develop some better applications of the beneficial microbes in the plant growth in different crop system. I think that's it. Hey, Meng Meng, you muted.
Oh,
You're muted.
all right. I want your attention to this my beautiful daughter here in this picture. When we were walking in the neighborhood, I just noticed this beautiful cluster of roses, yellow roses, and I wanna let them know. I said, hey, these yellow ones are really pretty, and smells really good, more fragrant than some of the others. So when I get close, this is what I noticed. On the bud, there's these little damages. So when I open up, one flower, this is what we see. A lot of thrips. You'll probably notice that, just lots of them. Almost every one of the flowers that's pretty much unopened or stuff, that they're just like gazillions of thrips. And then when I walked more blocks, I saw this one. So like, okay, let me check. That you'll see one or two thrips on this one, but not as many as the as this particular yellow roses here. And I was thinking, well, like in greenhouse production, we often use the sticky cards. To attract bugs, and mostly yellow cards, but I think for the thrips it's probably blue cards. So I want Erfan to take it from here and talk something about I asked Erfan, is there a color preference? Is there a color preference on the roses? The thrips.
Yeah, so it's a very interesting question.
Roses.
Yeah, for thrips specifically. And when we're talking color preference or just preference in general, when we're talking about insects,
Okay.
before you start staring at that image, there's a number of things to consider as an insect. If we're picking out food, for example, for us, it's not just the color of the food, but maybe also the smell, and also knowing about, is the food healthy or not, is optimal or not, or your cravings. For an insect, it's kind of similar. They look at multiple cues, and I don't know what this is. Just popped up here. Y'all can still see my screen? Check, check. Hello?
Yes, I can see your screen.
Oh, okay. I was like, oh shoot, did I just get disconnected?
Oh,
yeah.
What is that poll? Hold on. This is we're doing this post. Oh wow. 3/4.
Yeah, the poll just went full screen.
Okay, 3/4 of folks have seen the thrips in the landscapes.
Oh, all right.
Yeah,
okay.
Yeah. Okay.
So that's wow. I don't know if that's good or bad. That's
Well, this is definitely something that I have been hearing a lot from.
Okay.
So, yeah.
Okay.
So, hey,
yeah.
Hey y'all, if you haven't voted yet in the polls, click there. There's a little poll. So we just want to see whether you have seen, thrips in landscapes. Let us know.
Like within the last couple of weeks. Like
yes.
Recently,
yes. Within the last couple of weeks.
Not just like your entire life. Yeah. Yeah, so they use a number of absolutely. I think that was an important clarification.
I'm gonna count down. You have 5 seconds to poll. 5, 4, 3, 2, 1. We're gonna close it. Come on. All right, I'm closing it.
All right. So there's a number of cues, right, that they may use to choose what kind of plant that they might go to, right? And so one, with insects, a big one is olfaction, so smell. And they use their antennae, or sometimes they have little receptors on their feet or on their mouth or on their face that they'll use to essentially detect chemical cues and determine, what the quality of that particular host or that plant is. Now, in this case, if we're talking just visual cues, this is where things get kind of interesting, right? If we talk about what colors we see as humans, we are trichromatic, so we have 3 different color receptors in mostly in the red, blue, and green areas. And so we perceive those colors. Those are the only 3 colors we technically can perceive. And it's by our eyes mixing those 3 colors that it generates everything else in between, right? So it's mixing up those colors. Or something like a bee, you can see here it's pushed a little bit more towards the ultraviolets. They can actually see some colors that we can't see at all. So they would see, and we'll see here in a moment an example of how things can look quite different. And then you got things like hummingbirds, which are tetrachromatic. And I had to research this, there are very rare instances of tetrachromats in humans. It's like a crazy mutation where all of a sudden they can perceive, they have an extra bit of perception of an extra color that the normal population can't see. So it's essentially a mutation. But essentially these colors are, to be able to perceive these colors are ideally suited for what they're looking for, right? So if we go to a bee, for example, and what a bee might see, this is what we see in a flower, right? It might just be a solid yellow. And here are some simulated ideas, right? Because we can't actually see it, so we have to use certain types of lenses and filters that you'll see then. This is what actually looks like when you separate out UV color. It creates this kind of bullseye, which can attract the bees to that specific spot. So having that in mind, thrips at the same time, they are going to be able to see certain color cues that are going to be specific for what they are looking for. And if we think about what makes plant leaves green, what makes them reflect that kind of green color, it's because you have proteins inside that foliage that basically absorb colors on the extremes of the spectrum, and you end up with green and some yellow in there as well. And that's why that color yellow has always been thought as being a little bit attractive, because it's one of the colors that's reflected by delicious leaves. Now, if we actually go to some actual studies, so here's one example where they looked at the 3 different types of plants. They had mums, Matsumo aster, and Transvaal daisies, and they had different colors of them within a single cage. So if I actually go up here real quick, their design, right? They had like this large cage, and they had these are all the same species but different colors, and they release a bunch of thrips in there, and they basically count them and see how many get on the different ones. So for 2 of these, they actually had a yellow one, and you can see yellow is substantially higher when we're talking western flower thrips compared to the other colors. So that applies both to Transvaal daisy and to chrysanthemums as well. You can see it's about double in terms of number compared to all the other colors. They didn't have a yellow for aster plants, and it's kind of interesting that there's no differences between all the other colors within that plant. So then this was a study now looking specifically at roses, right? So your question was, if we want to really nail it down specifically on roses, yellow more attractive. So here's comparing a yellow rose to a different color rose. Each of these columns here, these groupings, we can see the number of thrips in when the flowers are about 25% open. You can see yellow compared to velvet deep red, there's quite a bit more in the yellow. Yellow compared to orange, there's quite a bit more in the yellow. Yellow compared to light pink, yellow compared to lavender purple, you can see they're a lot more similar. So there might not be a major difference there. But again, there might be more than just visual cues that are influencing this. And again, yellow compared to copper orange, a lot more in the yellow, and a lot more compared to bright red. Now, this is where it gets a little bit complex, right? So it's really easy to just try and bring it down to color, but it turns out that you might have some interacting factors, and it can be quite different between males and females, and that can be very important because males, when they go on a plant, will actually emit an aggregate pheromone or an aggregation pheromone to attract others to that plant. So really it's more important perhaps to ask, which ones attract the males more, and then from there you can basically build up a little nucleus population. So in this study here, one of the conclusions they found was that female thrips could discriminate different flora volatiles. So they can actually smell plants, whereas males could not. I would say that's the same between my wife and I. I can't smell anything, and she smells things that I swear not every towel apparently in our house smells. And so it's a use once and rewash, right? But both male and female thrips perform steady preference of flora volatiles under all 5 visual backgrounds. And we also find this where female thrips to the response of the female thrips to the floral volatiles was enhanced under green and blue backgrounds. So you have this case where if we're talking just females, that background color, green or blue, so in this case not yellow, was enhancing basically their attractiveness to that particular flower. So again, it might be also related to aromas. That's something to consider there. And now going to that last bit, and you ask, okay, so how does that relate to sticky cards, right? And so that's there's this an excellent colleague of mine, Sarah Jandrkic, who's based out of Canada, works in the Ontario Ministry of Ag. And there she had published a little article in Grower Talks, which you can find, where she basically compared different color sticky cards. So you got yellow, and you got blue, and you got this patterned blue as well. And what she found that consistently yellow traps collected more. There are some other studies that suggest that sometimes there's different companies all sell a slightly different wavelength of blue. And there's some that may work a bit better than the yellow, but it's not consistent. And they're finding, depending on where you are, so we're talking like latitude on the Earth's plane here, can affect how that blue actually reflects that light and thus changes this attractiveness. Whereas this yellow seems to be a little bit more consistent so far. And so the overall census, at least for now, is it seems as though yellow is still kind of a safer bet, but if you want to see whether blue would help you trap earlier and or more consistently, you may want to place some blue traps next to a few of your yellow and just make some observations. And you know, if the blue does work sooner and does work more consistently, so catches higher numbers, stick to the blue from that one company. Don't be switching around. Stick to that one blue, and for specifically for thrips trapping. So that's a hopefully a succinct answer, hopefully not too long, to your question of whether thrips are more attracted to the color yellow.
Well, thank you so much. Hey, Erfang, you know, to keep it under 1 hour, what about let's just, you know, stop right here, and then maybe let Becky talk?
Yeah, I didn't have anything else. That's all I was going to talk about.
Oh, great. Okay, Becky.
Yeah.
Thank goodness.
Thank you. Hey, and
actually, no, wait. I was going to do a movie review and zombies.
Give you a PhD right now.
Okay.
Becky, you have control.
Yay. I've got the power. Can you guys see my screen?
Yes.
Yes.
So I'm going to talk about two things pretty briefly. The first, because we're all kind of touching a little bit on some research that we've been involved in or are interested in. You know, depending on those of you that are watching, if you know anything about me, one of the big projects that I've been involved in the last couple of years is a USDA-funded SCRI project looking at controlling annual bluegrass, or Poa annua, primarily in turfgrass systems. So if you work in professional landscape industry, chances are you've had some run-ins with this particular weed. It's a winter annual, so we still see it out right now. There's some out producing seed in my yard as we speak, but it is going to start to wrap up its life cycle here as we continue to get a little bit warmer. So this is a $5.7 million grant, and we've got 16 different universities involved with it all throughout the country. So it's a pretty extensive grant, and we're kind of coming at this project from multiple angles. And so I thought I would just kind of give you guys a little taste of some of the stuff that we've been doing in Texas. And so we are doing sampling of annual bluegrass populations from 4 primary turfgrass systems: golf, sports field, sod production, and commercially managed residential landscapes. And we are separating those out across different USDA hardiness zones from north to south here in the United States. And of course, because Texas is so large, we have multiple hardiness zones that we're sampling from here. And so to date, we have sampled, it's actually a little bit higher than this now, but over 100 populations, primarily focused on zone 4, which you can see there, kind of central, and then zone 5, which is going to be further south. And so we're focusing a lot on that. And that's because a lot of our collaborators are already concentrated in zone 3 where our panhandle is located. So we already have some good representation there. So we have roughly 30 populations collected from zone 4 and 70-ish collected from zone 5. And we've done a pretty good job of sampling across each of these turfgrass systems pretty uniformly. So what we do is we go in and we collect about 20 or 30 annual bluegrass plants from different locations in each of these systems. And then we do a comprehensive herbicide screening to evaluate whether or not we see any herbicide resistance in these populations. And we're focused on kind of 9 primary herbicide products that are commonly used in turf. To just kind of see what does the distribution of resistance look like in some of these different systems. So that's something that's actively ongoing right now. We don't have a ton of data to present yet, but you can kind of keep an eye out for that. And then another thing that we did this year that was kind of interesting is we looked at Poa seedling emergence patterns, and we had one location in College Station and another in Brenham. And so we have these little 1 meter by 1 meter quadrants, and we collected some soil moisture and temperature data to kind of track that. Starting in the fall and throughout the course of the winter. And then we had a graduate student, of course, go out and hand pull little annual bluegrass seedlings in each of these square meter plots and do counts on how many seedlings we were getting on a every 2-week basis throughout the course of this past growing season. And so I thought I would share this because we found some kind of interesting things. So if you have a fall pre-emergence herbicide as part of your turf management program, you know that we typically recommend at least 2 applications a year, 1 in the spring and 1 in the fall. And we typically make that fall application when our soil temperatures drop to around 70 degrees Fahrenheit for 4 or 5 consecutive days. And depending on where you are in Texas, this can be anywhere from late August well into October. And this past year, we had a particularly hot and dry September. So we actually were able to push our fall applications off a little bit later. But we do that to try to create a barrier to protect our turf areas from the germination of these winter annuals at the beginning of the season. And this is kind of what our seedling emergence patterns look like for zone 4 up in College Station. And what we saw there is, we did kind of get that distinctive peak of germination and emergence kind of in the early fall. We had a few plants kind of come up a little bit later in the season. But as we go further south in Texas, we saw something that looked more like this. So this is more of like a bimodal emergence habit where we had an initial peak in the fall, and then we had a subsequent peak actually in January. And this is most likely due to the slightly more temperate temperatures and conditions as we go further south. And this is something that has been kind of suspected for some time, but we haven't had a lot of good data to support it. And what this really points to is, depending on the pre-emergence product that you use and the length of activity that we expect it to have in the soil, making that single application in the early fall may not be sufficient to protect your turfgrass areas for the duration of the winter. And so a sequential application may be critical for this particular weed to really create a more lasting barrier through those winter months, especially if you are in a more southern part of the state. Where we don't get quite as cold. Now, if you're using something like a spectacle, spectacle usually has a pretty long residual activity period, so we may not have to worry about this quite as much. But if you're using some of those more standard products like Dimension or Barricade or something like that, that's where we may explore the idea of doing a split or sequential application for this weed. So let me go out of this, and then if you want to know more about this research project, you can we have a website up. It's resistpoa.org, and you can read more about the project itself, some of the other scientists that are involved, some of the objectives that are being completed elsewhere. And so there's a lot of different aspects of this project, and you can learn more about it by visiting the website. And you can also follow our Twitter, which is managed by Dr. Jay McCurdy at Mississippi State, and he does a nice job of sharing some things. Last thing I'll talk about really briefly to make sure that Kevin has time to talk is a little bit about turf selection. This is the time of year where people are doing renovations and installing new turf in a lot of lawns and commercial areas. And so I'll get questions about some of the new varieties that are available and things like that. So I thought that I would just, for those of you that aren't familiar with some of these resources, just kind of share them with you. So one of the first ones we'll talk about is actually the Turfgrass Producers of Texas website. Some of you may already be pretty familiar with this, but if not, and if you are, if you do contract with growers to do sod installations, you might want to check this out. So this website has some nice information. One thing that it's got that I really like is a list of our commercially available turf varieties. It doesn't include everything, but it's pretty comprehensive, and just kind of depends on how much the growers report to the people that are managing this. But you can go through for all of these different varieties, and you can click on them and learn about some of the growers that produce them and where those growers are located. So, you know, it's kind of a useful tool for that purpose to kind of figure out where you can get certain commercial varieties. The other thing that I thought I would share briefly is this National Turfgrass Evaluation page. So for those of you that aren't familiar, NTUP is a program where they evaluate new varieties of turf when they're in the kind of the early stages of development, and they evaluate them in several different locations throughout the country to see how they perform just in those environments as well as under specific stresses. And they collect and compile all of that data, and it's publicly available. So it's a little tedious to go through. It just kind of depends on how much you want to nerd out on it. But for example, this is a file on Bermuda grass data from 2013 to 2017, and it has several of our newer varieties on it, including Tiff Tough, Tahoma 31, and Latitude 36, which are 3 fairly new cultivars that are very popular right now. We have a lot of acreage here in Texas. This right here is just turf quality data collected in different states. And it's pretty, it's very, very technical, very scientific. It's a 1 to 9 scale of how attractive the turf is, taken consistently by the same researchers. And the closer it is to 9, the better the quality. And so you can kind of go down here to, it says TX2, which is College Station. Some other files, it may say TX1, which is Dallas, and you can kind of get a sense for how some of these grasses are ranked and how they perform, both within Texas as well as across locations. So here in this file, we can see that Tip Tough was a very solid performer in College Station for 4 years of data collection. And so they have it, you know, these are all of the different Bermudas. They also have it separated into seeded and vegetated. If you go deeper into this file, they expose some of them to drought stress and provide data on that. So it's pretty neat, kind of handy little tool to have a more objective place to look at some of the information on some of these grasses. You know, of course, you can always go to the license holder websites for the grasses that are certified and still under patent, but sometimes you're going to get information that's going to, you know, mostly focus on the best things, right? Because they're trying to sell it. And so anyway, so just kind of a nice little tool. And if you have any questions about specific cultivars as you move into the season, you know, don't hesitate to shoot me an email and we can talk about it more, or comment on the Chat with Green Aggies Facebook page, and we can get into more detail next week.
Well,
okay, I'm going to pass it off to Kevin.
Thank you. I'm going to launch the next poll. So you guys have about 5 seconds to participate in this quick poll before I give the power to Dr. Kevin Ong.
Oh, are we done yet?
How's the poll looking?
I can't see.
Oh, Mung, you're on mute. We can't hear you right now.
Three quarters have seen the LeafSpot. So
Ooh.
yeah, Kevin, show your screen.
How do I show my? Never mind. I figured it out.
Come on, you're a pro.
Okay.
Kevin,
First
I have
things
to
first.
mention you have only 9 minutes.
9 minutes. Okay, I'm gonna take 5. We're gonna hit on some key points that are going to be important to producers as well as landscape maintenance folks. A couple of days ago, USDA confirmed Ralstonia solanacearum race 3 biovar 2. If you don't know what that is, that's a select agent. What does that mean? That's a bad pathogen to have. Well, they confirm it in Michigan, in a Michigan greenhouse on geraniums. And unfortunately, Texas is some of those plants were sent to Texas. And so if you are in the retail as well as wholesale and growing some of this stuff, just keep an eye on it. They are, if you have not been contacted yet, you will be, especially if you have plants from the Fantasia series, particularly Pink Flare. So keep in mind, if you have any questions regarding that, you can ask us through the chat or the question version, type it in there, or you can shoot me an email and we can discuss it offline. Short of it is, because it's select agent, it's one of those things that will be regulatory impact regarding this sort of finding. So keep in mind, if you do have this, realize that you may be contacted within the week. Let's see, what else do I got? All right, last week, I think Elfan showed some things with nematodes in it. Well, one of the interesting questions that came up during the week, was that you, Irfan?
Yes, sir.
Yep. Okay. So one of the interesting things that came up during the week was the question came up: Could this be transmitted or disseminated through irrigation water, particularly if there's a retention pond? The short answer to that is yes. In fact, irrigation water is a great way of disseminating nematodes, whether it's the juveniles, the adults, or the eggs. Now, however, we got to be real careful here. If the nematodes is in the pond, there's a good likelihood that it will drown. But what happens is when you do get runoff, the nematodes are going to be in the soil or and plant material. And so with the runoff that goes into, you're going to have the nematodes or the eggs flowing into that retention pond. And if that goes, they can stay in there for a short period of time, depending on the species. But nonetheless, if that irrigation water is being used and you do have that uptick and you do have that runoff that contain the nematodes, you can very easily spread nematodes to the rest of your operation. How do you deal with it? A cheap, simple way to deal with it is at your water intake from the pond. You might want to think about running it through a couple of sand filters. Again, not foolproof, but it's a great way of at least filtering out some of the nematodes, whether it's the adults, juveniles, or eggs. If you want to go a little bit more expensive and a little bit more, I guess you could say, a greater confidence, you can go with disc filters and all that. But remember, when you do something like that, you do have to keep up with the filters, check, make sure it does not get clogged. You could perhaps also consider some chemical injections. Some people would use chlorine. Some people use other types of disinfectant in the water. If you go that route, remember one thing: you need to be able to ensure that whatever material that you're using is not going to interact or create phytotoxic effect on your plants. So that's nematode. If you have any additional questions and no time to ask it here, remember, go to the Facebook page. What is it? Ask Aggie Green Experts or something like that. Type it in there and we'll address it next week. Let's see, what else do I have? Oh, right. So I have to show the cool picture of a rose that got chewed up by a bunch of insects. But more importantly, and this is going to be in the landscape, this is also going to be, if you are a grower and forgot to spray those darn things, you have black spots starting to show up right now on the more susceptible varieties. If you got those moisture and those cool, mild sort of temperatures heading towards the heat, you're going to have this black spot showing up on the various roses. The more susceptible ones will display it first. Now, if you don't take care of it at this stage, and by the way, this is in my front yard, I'm not going to take care of it because I'm interested to see what happens. What happens is the leaves are going to get infected and dropped off. If you're managing a landscape, if you see this right now and you have not treated it with a fungicide, this would be a good time to do so. Ideally, it should be done a little bit earlier, but what you'll be doing with the treatment is to protect the new growth from infection. And hopefully it'll get through this and get hotter and drier. We'll be out of the conducive zones. So the other thing that I want to jump ahead is leaf spots on woody ornamental. So I'm glad Mung had that poll out there. There's a whole bunch of different fungal leaf spots showing up, as well as bacterial leaf spots showing up on different types of plants. Just to quickly point out to you, if you do see water-soaked lesions, so it looks like an oil stain on a paper napkin that is pretty much captured by the veins, so very squarish instead of circle, that more likely would be a bacterial type leaf spot. And you tend to see that when weather's really humid and there's a lot of moisture available. We do see quite a bit of fungal leaf spots, everything from entomosporium on your photinias and your, was it the red tips, the photinias, the, what is the other one, the Indian hawthorn. We also start to see quite a bit of scospora popping up in other things like nandinas and so on. And part of it is because we have had rain in most parts of Texas. And what doesn't help in nurseries is a lot of the operations that are doing overhead irrigation. Couple of ways to deal with this is space out your plants properly to encourage air movement and drying out of the foliage quicker, and watering it probably early in the morning so it doesn't stay moist for long periods of time. Those things and in a production type system, also consider some form of broad spectrum fungicide in a program to maintain that you can grow plants that would look good and sellable. Last thing, how many more minutes do I have? 1. All right. So last week I think I showed you pictures of FireBlight. Let's go to here. Let's see. So if you have not seen FireBlight, make sure to go to the Texas Plant Clinic Facebook site. I believe I posted about FireBlight last Friday, and here's some pictures of it. So most of the FireBlight situation that we have in Texas is going to be on ornamental pears. You're going to see those tips dying. Let's see if I can pull one of these pictures up larger for you, and you'll see what we typically call the shepherd's crook. And there, we got it. There's a slight delay on that, and the leaves actually turn brown, almost black. These are typical symptoms of FireBlight. Occasionally, you'll see blotches on the leaves and so on. At this stage, to treat it would probably be not very useful, because this is a bacterial disease, comes in relative early, especially when the tree is in bloom. So when they have all that white flowers, probably depending on where you are, a month to 2 months ago, that would have been the time to spray. And the material that you would use this, they call it FireBlight spray. That's available for homeowners, but agromycin in terms of the commercial version, it is an antibiotic, it's streptomycin. So use it carefully. If you do use it, utilize it in a preventative fashion. But what you do is you look at the weather conditions, you look at the blooming period. That's how you time it. So this is one example where timing of a treatment would either give you success or no success. So timing of application is very important. into ensuring that you have a healthy looking plant. Last thing that I want to do. So this morning on the Facebook page, I posted a link to a YouTube video that one of my collaborators, a friend, sent me. And this was Mr. Ricky Bagnell with Sexton Bagnell and Sons, who's a greenhouse citrus producer. And so I got the privilege of taking a bunch of grad students out there last year, and he had this contraption called the disinfectant station. So Mong Mong is giving me the evil eye. If you guys are interested in disinfectant station, go to the Texas Plant Disease Diagnostic Live Facebook, check out that video, and I put it out there that if I can get enough comments, perhaps we can convince Mr. Bagnell to do a DIY how-to video to make one of those contraptions. All right, I'm gonna quit here. On time, on out.